start discussing our today's session. And if we see the part of innate immunity, what we have discussed till now, we have actually discussed a barrier immunity in detail. And last class we discussed about the gut microbiome. Today, my plan is to complete innate immunity so we can move to the adaptive immunity in the next session. Okay. So, as we know that we keep reading, sometimes things like do things at your own pace. A life is not a race. And sometime you get like before that I go, today's reference will be mostly from Harrison and Abbas textbook of immunology. There is very good article which Dr. Abbas has written, and it is doing rounds. Please remind me to share after our discussion. It's worth reading. It is about his total journey in the immunology, and it was so exciting to read this. And we have another category also, which tells life is a race. If you do not run fast, you will be like broken ender, right? So choose your path correctly, that where you want to go here or in the previous one. But that's all fine. Let's start discussing about our innate immune system and specifically focusing on pattern recognition, complement pathway, and cytokines. See, these were the words which actually discourage me to learn immunology, that whenever someone talks like this complement pathway cytokines, I was like nahi yaar, ye nahi padda hai, this is not my cup of tea at all. Okay, but then I realized ki if we just read, understand, and do not try to remember too much, life becomes quite easy. So do that with immunology, because understanding immunology will make your understanding of all the diseases so easy, specifically infectious disease and the chronic diseases, that you'll be able to correlate symptom and findings very easily. That is most important. As a clinician, if we can relate the process and findings easily, we can start doubting the results are wrong, or maybe it's a false or low calibration something. But if we do not have that clinico pathological correlation, life becomes too difficult. Then it's all reflex and robot like a treatment, right? So we'll start with the five components what we usually discuss with innate immunity. This is table from Harrison. One is pattern recognition receptor. We will take little in detail about this today, how what exactly happens at that point. Antimicrobial peptides, we have heard about this antimicrobial peptide like defensins, cathelin, protegreen, histine, leukotriene, and probiotics and all as a barrier immunity part. Wherever the mucosas and the skin and all parts are there, this antimicrobial peptide, because they are the first line of defense which are working. Then we have cells of innate immunity. I will discuss this, but not in today's class. Maybe when I'm discussing lymphoid organs, I will discuss about those cells. Then comes the complement system. Though classically taught, it's a part of innate immune system. Somehow actually it links innate and adaptive both, but we will discuss under this. This will be again a very important topic what we are going to discuss today. And of course the cytokine. So this three-part pattern recognition, cytokine and complement, we are going to discuss today a little more in detail. Okay. So when we start with our uh, pattern recognition, it is important to know that if we can recognize bacteria, virus, fungus, parasite, why I keep repeating this? Because I started immunology series with that, right? Immune response to infections. Because that's what, as a clinician, I can correlate very fast. I see viral infection, I see fungal infection, I see parasitic infection. I know the clinical manifestation of that, and then we started how body fights to that. Taking that as a prototype, the relation becomes quite easy to identify. So first, today we will start with pattern recognition and see how we proceed towards that. Okay. So the barrier is something what we discussed. There are epithelial barrier, then there are cellular barrier, then there is a gut microbiota, and then there are these chemical barriers with saliva, stomach acidity, and everything, right? So this all are fighting there. So there is a physical barrier and all to prevent this bacteria or the microorganism to enter. But once it enters, what happens? We are trying to identify it. This is very classical scene from Karan Johar's movie, right? So it is like just ready with that. The moment you breach it, we are ready. Where you are breaching it will matter. If it is extracellular, you have few TLR and the lectin receptor. If they're intracellular, you have node-like receptor, Reg-like receptor, and the certain CDs. And in endosome also, you have toll-like receptor and all. We have somehow got the glimpse of all this thing when we were discussing about the barrier immunity and all that. This is how the recognition is going to happen. There will be a lot of content which gets overlapped or we feel repeated. But in immunology, it's always better to repeat because we understand all the time better. So whenever the bug comes and it breaches the barrier, the next phase is to identify it. And for that identification, we need PRR. That's a pattern recognition receptor. The PRR identifies the part of bacteria, parasite, fungi, or viruses, and the part which is recognized, it's called PAMP. That is pathogen associated molecular protein, right? So the PAMP is recognized by the PRR, and then the further process starts. So this is the step one. When barrier is breached, those all PRR comes in the picture. Okay, so this is what here Harrison tells, that what are the types of PRR, what does it identify, okay, and where they are located. So first is toll-like receptor, then there is a Reg-like receptor, then there is a node-like receptor, and then you have CLR. Okay, so if you see all this part, in TLR you have a huge numbers, and each number where they are located on the membrane or endolysosome, and what does it identify. Now, is it really required to remember what does it identify? Sadly, answer is yes for MCQ. Otherwise, it is understanding that, okay, the bacterial or the microorganisms will have different component. The identification will require different machineries. So there are different receptors. The concept is quite easy. Okay. And for each microorganism, we have a different kind of killing system which works. So if we identify it, then we can activate that killing system more effectively. So that's why you need different kind of receptor to identify the structures and to regulate or to guide immune response to attack accordingly. Okay. So the TLR are not just limited to bacteria. Bacteria, fungus, virus, parasite, everything can be identified. Okay. Can you tell me what are the repeat MCQs of TLR? Always flagellin, TLR 5, which has been asked many of the time. TLR 11, because the examiner will think TLR 11 पहले नहीं बोलते थे. Previous Harrison Addison didn't had the TLR 11, तो वो पूछ लेते हैं, ठीक है. So it's a profilin like molecule is your answer, which is present in the protozoa, right? So we will come to this TLR in one more slide, but we will keep going as the time progress. Then you have Rig-like receptor, which is Rig1, MDA5, and LGP2, where they are located in cytoplasm. So what they will detect? Intracellular bacteria or viruses. Okay, so the answer is mostly the intracellular viruses, which are picked up by your RLR. Okay, influenza virus, where do we identify? Here, it's very easy. Fine. So this is what it happens. And then there is something yesterday, I think on group we were discussing that there was a case of dermatomyositis, and I wrote something called anti-MDA5 antibody, right? What is that MDA5? Here it is, anti-MDA5 antibody, right? So sometimes things are there right in front of our eyes, but we never think much about that. And then we read that anti-MDA5 antibody dermatomyositis, or which is amyopathic dermatomyositis, can be triggered by virus infection, specifically RNA virus infection. This whole line makes lot of sense. Oh, this is what it happens, right? Even there was kind of prediction or the words given by immunologists during COVID that it can precipitate this kind of syndromes, and many of the cases already have been reported that after COVID there is a transient dermatomyositis which completely resolved after some time. Okay, so this all can be triggered. These are the initial stage where it starts. Okay, then you have CLR, and in that specifically, they are more for fungi, right? Dactin, Dactin 2, Minkle. And someone will tell, oh, we have heard all this kind of Dactin and Minkle and all when it comes to the fungal infection or all, right? So these are overall working. Node-like receptors are also in cytoplasm. So intracellular viruses are catched by Rig-like receptor. Bacteria are caught by node-like receptor, right? So intracellular bacteria, those roles will come for these things. Okay. So this is overall recognition. I know it's a very hectic slide or busy slide, what we can say, that the one time you have the receptor, then where they are located, what are the ligands, what they identify, and where they are present. So ultimately, you have a lot of organism which expresses something. This ligand is nothing but the PAMPs, which once entering on the body gets either inside the cell or outside the cell, and it is identified by like this. Okay, so if we start this table in this direction, life becomes quite easy compared to starting in reverse direction. Okay, this is what we can understand, that a bacteria came or the virus came, it expressed this ligand or the PAMP, which actually was present at the level of cytoplasm or the site or the cell surface or in the endosome, which got recognized by the body's receptor like this. So then it becomes easy. And that's only my job, that information is given, but how we actually make it little easy to interpret for all of us. Okay, so we have just started pattern recognition. We will slowly go into this. Now let me make this table little more easy with this diagram. Okay, so this is just explained by the TLR families. Okay, so if you see in this TLR family, TLR1, 2 in pair, TLR2 in pair, TLR4, TLR5, 2, 6. They identified either bacterial lipopeptide, peptidoglycan, LPS, flagellin, or the lipopeptides by this. And once it is identified, they are at the cell surface level identification. Sometimes if they are intracellular or the endosomal level, which identifies the viruses by identifying DNA and RNA, right? We have DNA virus, we have RNA virus, and it gets identification in this structure. Okay, so this is how it gets identified. But the important point is, once identification happens, what next, right? So now the microorganism has breached the barrier, has entered into the body, and has been recognized by the PRR because it was expressing PAM. After recognition, we have to act, right? So the intracellular signaling should start, and that is the signaling pathway, right? So once that entry happens and Jaya Bachchan calls, but after that, that signaling happens and Shah Rukh Khan gets bitten little, right? So this is what happens. Once the recognition has happened, if you see the downstream signaling through MYD88, TRIF, and all things goes into cytoplasm, which goes to the nucleus, and nucleus, it will affect the gene expression either through NF kappa B or IRF. This IRF is mostly the viral response where the interferon gets activated and the antiviral state is established. While your NF kappa B is more with the activation through bacteria, fungus, and all, which will lead to acute inflammation, and it will also stimulate the adaptive immune system. So now the link starts getting, okay, no, we identified, and now we are doing signaling, and this is the final result or action we are getting in the picture. Okay, so this is the role of pattern recognition receptor, that whenever a microorganism comes, it gets identified according to its PAMP and its location established. And after that is activated, the signaling pathway comes in the cytosol, which ultimately sends message to nucleus, and it will change the transcription or activate the transcription factors and all. And there will be the expression of pro-inflammatory milieux according to what has actually attacked. If virus, then interferon dominant, and if it is more of fungus and bacteria, it will be more of NF kappa B pathway dominant things which will happen, right? But this is required, right? If there is something to be killed, if the virus is to be killed, this has to be expressed. Now, if there is some mutation or the loss of function of mutation in the interferon genes, the virus response won't be that great, right? Antiviral response won't be that great. So then there the theories came that few people are quite susceptible to COVID because they do not have this thing, so they do not develop the antiviral state, and the virus persists for a longer time there and does more damage to them, right? Because we saw in a massive scale, so we could imagine all this thing from the data, and then later on it was proven to the certain degree, okay, if these are the genetic defect present, ultimately life was going fine, but if this kind of infection comes, the fight might be little less. Okay, so this was the pathway there. So, and this is what we were talking in little more detail if we go for the TLR. So whenever TLRs are activated, either here or into the endosome, it will activate either the IRF3 pathway or the through MYD88 IRAC pathway, and then ultimately you will have the response of interferon related or this. Do not get at all depressed by all this kind of images. These are easy to interpret. The important thing is not to remember much, not too much. Okay. We have to just remember principles that body has to act in that. And then slowly, when you will start going in detail of immunology, if you are going towards that direction of becoming ID specialist or immunologist, there you will get the question, if a patient has a IRAC deficiency or the IRAC related mutation, what patient develops? If there is NF kappa B related things, what syndrome patient develops? So these are all our inborn error of immunity. Okay, so this is a later talk. Someday we might discuss all this thing, but today we'll keep it very simple. Today it is more about principles to understand, and for that I cannot take shortcuts. We have to actually show what is there and then try to understand that. We won't go in that much detail even. Okay, so after recognition, this is the part what happens. So this is what that everything is done, but tell me something clinical, because all these things are too much that you are telling there is a microorganism which has breached the barrier through PAMP. The PRR recognized that depending on location, the signaling pathway started in cytosol, then the genes got activated, and now we are acting to kill or to clear that microorganism. But how does the body actually reflect, or how does my patient tells the problem is there? So now remember your septic patient with sepsis or SIRS, and then try to see that. See, our first year residency was all mostly the spinal reflexes, right? If these are the things, this is likely diagnosis, and this is what antibiotic I'll start, these are the investigation I will send. So what we have seen mostly in the first 6 months without thinking why it is happening is this picture. Okay, so whenever the innate immune system is activated through PRR and the PAMP and all and the signaling pathway, there is release of cytokines like TNF, IL-1 and chemokines and all, which will lead to all leukocyte activation and all, which will lead to increased permeability and all. So at the site of infection, you will see there is a redness, there is a swelling, there is a pus formation, everything which can be seen as a local inflammation. If it's pneumonia, you can see patch on the x-ray, you get the crepitation on your auscultation. This all are features of local inflammation which is active. Now, along with that, what does this cells do? They secrete cytokines. We will come to cytokines soon. So now this cytokines has effect on different organ. We get fever, right? In sepsis, we get fever. So that is through the TNF, IL-1 and IL-6 effect on the brain and further thermoregulatory pathway. When IL-1 and IL-6 affects liver cells, the acute phase reactant gets activated and you get high C-reactive protein. Yeah, it makes sense, right? So there it comes from that. It affects bone marrow because there is a problem. So we need more cell to fight. So it's a leukocyte production, and you get leukocytosis, increase in TLC, and the neutrophil will increase. It will be little premature, so you get the band form of cell also, right? So all makes sense that once that pattern recognition happen, what ultimately we get. It also affect the heart, and we say, oh, output is low, septic cardiomyopathy, right? That is a result of this. Then there's endothelial dysfunction and all, which may lead to DIC and the capillary leak, right? And then there is a myalgia and insulin resistance and all part. So we have seen this in clinic. We just do not think sometime why all these things are happening. What are the inside molecular mechanism all things are happening? And some people will ask me, is it really required to know all this thing? I'm not sure about the answer, but I would always like to know what I'm treating and how things are happening, because sometimes it does not match. That's the case. You make a difference. You know, there is something problematic. It does not look classical, and then we have to keep thinking what could have gone wrong, and we can actually help patient, right? So this is what we see in clinic as a total result of innate immune activation, the patient episode of sepsis or septic. There are a lot of other components, but this is very basic, we can say, which explain lot of symptom, right? Sometimes we come out of the ward and relative will ask that why still is having fever. We cannot tell TNF, IL-1, 6 and IL-1, right? but we understand, okay, why still she or he is having fever, because we have not exactly caught the trigger what is doing all this activation. The day we got trigger, we hit it and the fever goes down. We all see this. Okay, just we have to start correlating this part. Okay. uh, the innate immune system has many functions. Phagocytosis, I am not going in much in detail. We have already discussed it in the two classes, once with the barrier immunity and once with the other part also. But as we know that phagocytosis to happen, you have your cells like neutrophil, macrophage, and all, which will identify the bacteria or the fungus, whatever it is, through its receptor, scavenger receptor or Mannose receptor. Once it identifies the bugs, it will take it inside. Its endocytosis will happen. Then it will be the phagosome, and then phagosome and lysosome will bind to make a phagolysosome. And in that, there will be the reactive oxygen species activation, oxidative burst, and the bacteria will be lysed and cleared. That's a phagocytosis, what we see in our picture with the innate immune system activation. There are a lot of things which happens with innate immune activation. One of them is phagocytosis. We have discussed that. Even we have discussed inflammasome activation. I'm not going to go in much detail because I'm waiting for the complement, which is one of the main thing to know. And once this phagocytosis happen, again the same thing, it can actually enhance the adaptive immunity. So we have always been taught that this is innate immune system, this is adaptive immune system. Believe me, they are not two different systems. They are same kind of continuation. We are trying to understand. For our understanding, we have divided this part, right? Otherwise, once the bacteria attacks, the innate starts kicking in, but it has to tell adaptive because the better and the specific killing is done by adaptive things. So everything is in the link, but for understanding, we make it little separate and we take separate classes for these things. Otherwise, things are quite linked. Okay. Now that is the part that whatever phagocytosis or the pattern recognition happens, does it actually touch adaptive at all? The answer is always yes. Okay, so all through this TLR, CLR, or the not like receptor or the Mannose receptor, somehow we touch this system, and that we will discuss sometime later also at how it is working. But our concept wise, it should be clear. These are all from Harrison. I'm not bringing anything about that. Okay, this is all given in our Harrison, that it touches the adaptive immune system also through this pattern recognition receptor. Fine. So this is the part. Now, this is a very important slide, I would say, that if we want to summarize pattern recognition receptor with a movie of Karan Johar, this was the first scene, right? The Shah Rukh Khan started coming to home. So it was the PAM. It was trying to breach barrier with its pathogen associated molecular protein. Okay. So who welcomed the pattern recognition receptor, right? So that was, okay, I will identify you, and then the worst thing happens, the signaling. Okay. And then the ultimate outcome. This is what we learned in last 10 to 15 minutes. That when pattern recognition happens, this is the ultimate outcome we are going to achieve by taking care of the attacking bacteria or virus. So this is the first and very important concept to understand. Where the barrier immunity fails, this is the part which starts coming in picture and plays a very important role. So I hope up to this it is clear. It should be clear. And we go to our next part of complement pathway. Okay. Now, why it is important to discuss complement pathway? Because it comes to exam as a final short note. That's the first explanation. There are so many diseases related to complement pathway. This is the second little better explanation. And third, very important, there are certain therapies which are targeting complement pathway. So we all know something about complement, but not much in detail. Okay, so I will keep it to some 10 to 15 slides where you can actually complete your short note very perfectly, and then we will see how we proceed. But before I go there, can someone just type in chat box, do you know any drugs which target complement other than eculizumab? I know eculizumab answer was ready for PNH, which targets at the C5 level. Okay. Do you know any other drug which works at complement pathway? You can type in your chat box and we will see as the class evolves. Okay. So if we understand the complement system, I'll put some question. I'll get little slow. I really need, even though we are now just 13 or 14 active in the class, I really need to know some answers, that if we have much idea of this or not. Okay. So if we ask about complement system, who discovered it? I really don't want that answer perfectly, but it was the Jules Bordet who initially gave name Alexin, and after that Paul Ehrlich named the term complement. Why it is term complement? Because it complements bacterial or the parasitic or the fungal killing. That's why it is called complement. Okay. And now this is very important, that how this Jules Bordet got this, that there is something additional also. So initially antibody was discovered and we were ki chalo antibodies killing the bacteria. So whenever we were mixing the serum which has antibodies to the bacteria, the bacterial activity used to go low. Okay. But when we heated the serum, that activity, lytic activity came down. So then this person thought, oh, there is something in the plasma which is heat labile, which actually helps antibody to kill bacteria. Help means it complements antibody to kill bacteria. That's why it is called complement system. Just 1 second. My laptop battery is little low, so I'll just fix the charging and we will continue. Okay, so this is how the complement actually came in picture. So what we recognize, one property of that, it's a heat labile plasma protein complementing or helping the activity of antibody to kill the bacteria. That's why it's a complement, right? So it is very easy to understand when we really go into the past history and see what is actually happening with this. Okay. Okay, fine. So this is the second thing. Now the third part is what are complements? We know now. It's a hit labile uh, proteins that assist in killing. Okay, now the next question comes, where are they? They are in plasma in inactive state, means they are regulated. Right now they are there in our body, but they are regulated, that they are not active. If they are active, we will have the features of inflammation. How we get it, we will come to that. How many we know? 30 is at present number, but many we might not be knowing. Where are they synthesized? In liver. So in liver diseases, complement level might come low despite the things are fine, because we will keep sending this complement level C3, C4 and all in autoimmune diseases. But the basic things are very clear. We should know that it is majority produced by liver, and to the certain degree, they are also produced by monocyte, tissue macrophage, and epithelial cells of the GI tract and the GU tract. Now, one important question comes, that if we are doing a serum analysis, we do total protein albumin, right? And once we delete total protein minus albumin, what we get is the globulin. In this globulin component, actually complements are hidden there. Okay. We do separate analysis of C3, C4, but as such, they are plasma protein related to that. So when we do TP minus albumin, the ultimate part we get is globulin. We all are taught this, all are antibodies, right? IgG, IgA, IgM, IgD, and IgE. But there are certain other component which comes in the portion of total protein. When we exclude albumin, is not just antibody, but there are also other proteins, including your complements. Okay, so they are where? In serum. So if someone says that you have to send complement level C3 and C4, which tube you are going to use in your clinical ward? Plain serum tube, because it is there. Okay, sometimes the specific things of or the basic concept really helps us. Because when I was asked first time you have to send ANA, I was not sure how to send it in EDT or in the plain serum or what, right? But then it's antibody, it's a globulin part, it should be sent in plain serum. Over. Answer is very simple to that, right? So they are majority synthesized in the liver. Activation and function, we will go through this part in the next few slides, and clinical implication of complement pathway. So if you get a short note on complement, and if you can answer this 5 question plus this 3 question in above slide, I think life is quite easy. Who discovered it and why they are called complement, and what are this complement? This is your introduction. Then you go for the next step, that where they are, how many they are, and where they are synthesized. And then you go for activation and function. We'll go in the next slides. and then the clinical implication of that. If you can write this 8 label, your complement system short note is ready. Okay, so now just let's work for this two part before we come to or jump to cytokine. Now do not get intimidated anyway by this pathways and all anything, but there are three pathways. One is alternate pathway, one is classical pathway, and one is lectin pathway. So for any pathway you need a starting. Now this alternate pathway is something which is a fluidic pathway. They are constantly there in the water, and water is one of the activating factor in that which plays a role. Okay, so it happens on the surface of microorganism. So on the microbe surface on these things, this activity starts taking place, and ultimately it makes C3 convertase, that is C3bbb. Okay, that's a 3,3 convertase of the alternate pathway. For classical pathway to activated, to get activated, you need antibody. You need antibody, means adaptive system, right? So when we were discussing about this part, that innate system and complement, but now you are telling me to activate something of innate, you need something from adaptive, means everything is linked. That's what we have to start remembering, that complement system also complements adaptive immunity, ultimate aim to kill the bacteria. And this is how they were discovered. They're helping helper to antibody, means to adaptive system. So it is easy to accept it right now. Though Harrison told it's in table, it's a part of innate component, but in a broader understanding, everything is same. Okay, it is like it is linked to everything what we are looking at. Fine. So this is basic understanding to have. So now you can see this is bacteria. On that, this is some kind of antigen which are getting caught by this Y-like structure, and this Y-like structure is nothing but the antibody. Once antigen antibody complex is formed, it is getting recognized by C1, which is starting of classical complement pathway, specifically C1q. C1q is the starting of classical complement pathway. Once that comes, then you get activation of C4 followed by C2, and then you get C3 convertase, which is C4b2A, which is a C3 convertase of classical pathway. In lectin pathway, the starting is by mannose, which are specifically present on many of the bacteria and many of the fungus. So through that, MASP1 and MASP2 get recognized through the mannose binding protein, and then the pathway of C4, C2 gets activated, and you get C4b2A as a C3 convertase. The C3 convertase sometime used to be MCQ, but I have not seen much in the SS level MCQ. From MBBS to MD, we used to get this, that for alternate pathway, what is C3 convertase? For this pathway, what are C3 convertase? So the common goal of all this pathway initially was to make C3 convertase. Once the C3 convertase is made, it will actually convert C3 into C3A and C3B, right? So this is C3B and C3A. Now C3A has a definite role to play. We will come to that. And C3B also has a definite role to play to take the pathway further to make membrane attack complex. Okay, so if we go to the next step, this is the next step. If you see this, through C5 convertase which happened after C3 convertase was there, the C5A get activated. Again, it has a role to play. So C3A and C5A will have its role in anaphylaxis, inflammation, and all this part, and then things starts coming in the picture. But once this is getting established on the cell surface, C6, 7, 8 joins, and the C9 will make this tubular structure which will make the pore on the bacterial wall, and this bacterial wall will be broken because of the total imbalance there. So this is how the complement system is going to kill, through this pathway followed by this. So what we understood, the C3 convertase is the ultimate goal to take, and after that C5 convertase takes place, and then the cell lysis takes place, and the rest of the message is getting conveyed by C3A and C5A. So ultimately, three important effector of complement system are C3A, C5A, and membrane attack complex. If these are the three parts affected, the problems becomes too much. The ultimate starting of complement pathway is either the bacterial surface where the alternate pathway gets activated, or the antibody getting identified by C1q where the classical pathway is getting activated, or the mannose, which is a polysaccharide on the cell wall, which activates the lectin pathway. That's our starting point. This is the common point where they converge. This is the ultimate outcome, and the effectors are C3A, C5A, and membrane attack complex. This is how the killing happens. We have not discussed much about C3 and C5A. In next one slide, I will go to that. Okay, but I hope this is clear that how does it start, how it propagates further, and how it kills. Okay, how it kills the organism completely. So now we will go to this part of C3b and all. That C3b, whenever it is there, it will help in phagocytosis of microbiome by the opsonization, right? It will make it surface ready that other things can come and kill it. So whenever a microbe is coated by C3b, it can be easily identified by these cells and can lead to phagocytosis of microbiome. Second is the stimulation of inflammatory reaction, where you get the predominant role of C3A and C5A, which will actually lead to recruitment of other cells and all, and will lead to destruction of further microbiomes by this pathway. But these are the things which are actually getting into the result. And third, what we told is a complement mediated cytolysis, where the membrane attack complex will come and will do the osmotic lysis of bacteria by making these pores by the C9 as a final product in the category. Now, see, ultimate aim was to make this pore, but to make this, you need complete machinery like this. And for that, you need starting like this. Okay. So this is how the complement system totally kills and it does its function. Now, Harrison has done it a beautiful summary of this. Your three pathway. One was your classical activation pathway, which gets activated by antigen antibody immune complex, which touches the C1q. After touching the C1q, C1R and S gets activated, which ultimately activates C4 to C4 and then C2, leading to C3 convertase, and C3 gets activated. In that, C3b component will go further, will activate C5, and in that C5a go little out, and C5b will go further to make the total poly C9 a pore, and the membrane attack complex lead to lysis of the bacteria. Meanwhile, all these things are happening. The C1q help in clearance of apoptotic cell. I always used to ask when I used to discuss phagocytosis that whenever we start killing the bacteria and all, and the cell is also getting lysed with that, what happens to that? It gets cleared. Okay, suppose the cell is not getting cleared, the apoptotic cell is not getting cleared. What happens? Your DNA, RNA, protein, everything is exposed to immune system, which is native, but they're exposed. When the native things are exposed, what body does? It makes auto antibody. So now when you read lupus or any other autoimmune disease, you will see if C1q is defected, there is a chance to develop autoimmunity. This is how the autoimmunity develops. So if we start developing concept, it becomes beautiful. Okay. So now this is what we were talking about C1q. Now second pathway was mannose binding MBL pathway, where the microbes with the terminal mannose group will express that, and then C4, C2 get activated, and the further story goes on. And the alternate pathway which takes place on the surface of the cells, and then the ultimate story gets activated by that. Now we talked about C3A. So C3A is anaphylotoxin. In anaphylaxis, it will help opsonin and lymphocyte activator. And C5 is anaphylotoxin, right? So now whenever we read about this anaphylotoxin, a disease will definitely come in picture called hereditary angioedema. Okay, so that is what C1 esterase inhibitor deficiency. So then C1 esterase inhibitor is not there. Their C1 is continuously active. When they're continuously active, it is continuously activating C3 and C5, which is giving anaphylaxis like reaction. Oh, now it is easy to understand that how C1 esterase inhibitor is giving all this part. And then we actually infuse C1 esterase inhibitor, all this hereditary angioedema episode starts coming down. They're available in market now. Too expensive, but they are available. Okay, so this is how understanding things actually start helping us in the practice. Though this is a simple summary by Harrison, but if you understand all this part, this is the ultimate outcome of this. If you can write this much, life is quite cool with that. Okay, so this is the complement pathway. These are the function of complement pathway. Two slides enough for till now what we have done for the complement. Okay, now we'll go further. What are the complement associated diseases? Because we are doing short note, right? Complement pathway, then complement function, then complement associated diseases. So if you see in classical pathway, if these things are affected, C1q or the C1R or the C1s or the C4, what happens, you know, now the clearance won't be good. So you will develop autoimmune diseases, right? The immune complex syndrome. And you will not be able to kill bacteria effectively. You will develop pyogenic infection. So that's an easy answer. Same happens with the C2, immune complex syndrome, and few will present with pyogenic infection. If C1 inhibitor is affected, you will get immune complex diseases with pyogenic infection. Then comes the C3 and the alternate pathway. Again, you will get this. But now, whenever you have factor D, properdin I or H affected, it will lead to more of Neisseria infection and the pyogenic infection. Properdin necessary for Neisseria infection is quite an important MCQ for us. And where membrane attack complex is affected, we cannot make the pores, right? The C9 cannot make the pores. So Neisseria infection happens with that part. Okay. And that used to be a very common MCQ. Disease of membrane attack complex, patient is susceptible to what? Neisseria infection. Now the question comes, every bacteria has wall. Then why Neisseria wall is affected more? Any thought on this? Okay. See, every bacteria requires killing, and that can be done as such with so many mechanisms. Neisseria bacteria compared to other bacteria has thicker wall. So they have definite dependence on pore making complement terminal pathway with membrane attack complex. If they are not there, it is very difficult to kill Neisseria. That's why when the membrane attack complex is affected, there is a high susceptibility chance to Neisseria infection. We have to keep giving thoughts all the time and keep asking why, then life becomes quite easy on that. Okay. Now tell me some more disease. Anything more we know about this? So this is a very good article what I read for the complement and related targeted therapy. They have given the complete list there where complements are associated. We will run through it very fast, but few are must to remember where complement primary dysregulation lead to problem, which is hereditary angioedema, you know, PNH, atypical HUS and TTP, thrombotic microangiopathy, C3 glomerulopathy and MPGN, and transplant rejection. Okay. Then comes certain autoimmune diseases like NMO, MS, GBS, myasthenia gravis, lupus, IgA nephropathy, RA, Crohn's, ulcerative colitis, autoimmune hemolytic anemia, pemphigus, and antiphospholipid syndrome, where this complement has definite role. Then comes other inflammatory diseases like this, and then degenerative disease, and then the acute injuries where complements play important role. Means it is one of the pathway which is linked to innate and adaptive, so it can have multiple association with the diseases. This is very common and important to remember for us in the MCQ. This is from Harrison, right? But this is overall understanding or concept to develop that it is not just limited to few diseases. The complement system is everywhere. So it actually can impact many of the system which are located there. So I've got one answer, which is pegcetacoplan. Okay, fine. So that is one which works on the complement. We will go to the further drugs which works other than eculizumab, and this is a total list. And as you told that this is C3 inhibitor, which was talked too much in the NEGM. And against that, you have eculizumab, which is C5 inhibitor. And then you have zilocopan, which is C5 inhibitor. And very important, avacopan, which is there in your ANCA vasculitis, which is C5 inhibitor. So now these are the drugs which targets complement. So now you know very basics of complement, that how they were discovered, where they are, how many they are, where they are synthesized, and then going further, if we go, what are the pathways of complement? What happens as a result of complement activation? What happens if complements are disturbed, the disease associated with that? And what are the complement targeted therapies, right? So this is the perfect short note to write in your theory exam. You know almost everything about complement now. These are additional information. I'm not going much into that. But complement also has to work on the receptor, and this receptors are located in almost all immune cells, macrophage, and the B cell, which are complement receptor 2, 3, 4, and 1. Okay, we'll not go much in detail, but as we should just know. Then I told that one state that they are located in the plasma in inactive state. They are inactive state. So there must be some regulatory factors which are keeping them inactive. And what are these regulatory factor? Again, a busy diagram. Not to worry, just look at the gray part. C1 inhibitor is natural inhibitor to the complement part and all in the C1 part. So this is the regulator of the classical and the lectin pathway. When C1 inhibitor is not there, you can imagine this is completely active and patient can get hereditary angioedema. So for treatment, if you supplement additional C1 inhibitor from the outside, you are done with your job and life is good, right? This is how it works. Then the second, if you see the gray, complement receptor 1 and decay accelerating factor, which are at the level of C3 convertase. They are natural inhibitor of that. Then you have C4 binding protein, which is at this level also. Then you have MCP, factor H, and factor I. And then you read factor H, and all are related to PNH kind of etiology and or the HUS kind of etiology, and life becomes quite easy. Why they are related to this? Because they are regulatory. If they are not regulating, the complement is constantly active. When they're active, they are doing lysis, then you are getting hemolytic anemia, you are getting microangiopathic hemolytic anemia, right? So it is sometimes makes lot of sense when we start understanding. We only get screwed when people start making MCQ out of that. Okay, but we cannot stop them. And we also have to go in that process to get up to some level in India, right? So this is important if you are just in the journey of that. Others understanding wise, there is no limit that you can't understand any of this what is given in this. Okay, so this is all about your complement pathway. I hope there is some energy. I just need 10 more minutes for cytokines, which we have ignored like crazy. Okay, we all have ignored cytokines. Who reads this much? And I'm going to ignore it. Okay, just give me 5 slides and I'm going to close it there. So till now, what we have done is we are today on completing session of innate immunity. We started with the pattern recognition. We completed complement pathway. We are little exhausted. If someone can send me score, I'll be very happy what is happening in the match. And then we will still discuss the cytokines. Okay. So before we start, the question is very simple. What are these cytokines? Cytokines are molecule which we never understand. Okay, they can come from many of the cells and they are kind of messengers. So let's go little in the detail of that. Okay, so we'll come to this part, but before that, just see the definition of the theory part of the cytokine before we come to that part. They are soluble protein produced by wide variety of cell types. So we can tell, though we are discussing under the heading of innate immune system, it can come from adaptive for sure. Okay, so there is no limit of that. We have to make that part. So it's critical for both normal, innate, and adaptive immune response, which is involved in regulation of growth, development, activation of immune system cells, and mediation of the inflammatory response. Means they are required. Otherwise, message cannot reach from one cell to another cell. They function in autocrine, paracrine, and endocrine matter, like hormones. It functions in all. And then you realize few hormones are actually cytokines and they work in all this manners. Okay, so the things are not as easy as it looks, but they are okay to understand. So the basic role of cytokine is this: we had like Shah Rukh Khan coming, inducing stimulus, the microbiome and all whatever we say. It actually stimulated the gene from cytokine gene production. Then we had TNF, IL-1, IL-6. These are cytokines. So cytokines came out of the cells. It worked on the receptor of that cytokine, again led to signal gene expression on the target cell, and we got the biologic effect like fever, like inflammation, like a swelling, like pain. All these things are result of cytokines. So what you get, SIRS or the sepsis syndrome, is nothing but the biological effect of cytokine which get activated by the stimulus. Okay, there are a lot of things, lot of complexity. What are the key properties of this cytokine? Before we go to that, in COVID and all, we have heard this term cytokine storm. Before that also this term was there, but not commonly heard. We had only few diseases where we used to discuss cytokine storm and all. Okay, but now it is crazy. So this cytokines, it becomes too much. can lead to too much high CRP, too much high fever, organ failures, and everything. And they can come by activation of this kind of cells. And this all were somehow affected in many of the infections. If they're overactivated, the cytokine storm comes in the picture. That's a general picture or the clinical implication, what we can say about the cytokine. What are the properties of this cytokine? They are quite flexible, quite flexible. So one is the pleiotropy. Cytokine is just IL-4, but it has lot of role. It can activate and differentiate B cell. It can work on platelet. It can work on mast cell. So one thing, multiple function. That's a pleiotropy. Statin has multiple effects. So we call pleiotropic effect of statins, right? So that's the first quality it has. The second quality what statin has, sorry, what the cytokine has is redundancy. So many cytokines different, but the same function. The B cell proliferation can take place through this. That is actually a quite organized system where one thing is affected, other is taking care, right? That is what it is trying to tell. It's a redundancy. The third is synergy, that IL-4 and 5, if they are together, it activates B cell switching to IgE more efficiently rather than single thing working. So that's a synergy. Fourth is antagonism. They can oppose each other, right? IL-4 and interferon gamma. And fifth is cascade induction, what we already saw. That result of one can lead to result to other, and the total body response can come. So when we are talking about cytokines, the first what we talk is what is basic concept of cytokine. Then we talk about cytokine storm, what it can lead to if things are activated. Then we talk about properties of cytokines. Then we talk the families of cytokine, and here we do not talk much. We get to stuck here in that Anant Narayan book of microbiology. ये chapter से आगे नहीं पढ़ा जाएगा. This is done. Okay. But now just we will go through. There are certain families for MCQs of cytokine. That one family is interleukin 1. Then there is a class 1 cytokine family. This is a hematopoietin family. Class 2 family, which is interferon family. Then there is a TNF family, 17 interleukin 17 family, and then there are chemokine. So chemokine are part of cytokine. Chemokine are part of cytokine. Fine. So now if you see the numbers of this family, members of this family, you will find few things: GM-CSF, G-CSF. Oh, they are cytokine. Yes, they are cytokine. Okay. Growth hormone is also a cytokine. Prolactin is also cytokine, which comes in the hematopoietin class 1 cytokine family. Okay. So sometime if someone is really barbaric, can ask MCQ. All these are part of cytokine 2 family, except, okay, be ready for that if you want to go to immunology for some time. Once you go to immunology, forget it. It's fine because no one asks after that. Okay, before that, anyone will ask. Okay, so then you have BAF receptor, April receptor, and all, and which again comes to the cytokine families of the TNF family, right? So these are the few things which becomes. Now, almost on many cytokine, there are drugs, and this is again a boring thing which people keep asking, that these drugs target this cytokine or this cytokine receptor, but let it be, it will come and we have to be ready for that. Okay, for MCQ for sure. In practice it will become easy because you'll be expert of that once you will be able to practice all those things. Okay, so that won't be a much matter, but this is the cytokine family. Now to identify the cytokine family, you need a new heading called cytokine receptor family, because you have so many cytokines, you cannot make so many receptors. So you make a group of receptor, the few cytokine will receptor will recognize few. Now see, the problem is so much, it is not like cytokine one family is all cytokines are recognized by similar receptor. They can vary. Okay. So this classification sometime was on the discovery, that how they are getting discovered and they are getting named. Okay. So it is so haphazard. That's why I do not remember. Just understand and leave it. Okay. Now these are the cytokine receptor family. The first is type 1, type 2 cytokine. Then there are TNF receptor, then there's TGF beta receptor, toll receptors, chemokine receptor. All everyone has seen this structure like G protein couple receptor family. And then there is a receptor tyrosine kinase. Okay. So now we know a little about cytokine. They have family, their receptors have their family. It can go into the storm, and there are certain properties of cytokine we know. Now two more slides and we complete this part. Once again cytokine is activated, suppose this is a cytokine activated, it will binds to its receptor. After binding to its receptor, it will lead to cytosolic activation of the pathway like JAK-STAT, which will lead to the nucleus transcription and will lead to the effector protein, which will get released from the cell and will do its effect like fever and all immune response what we are getting. So ultimately, a molecule which is stimulated came from a cell, attached to its receptor, activate the cytosolic signaling pathway, led to the transcription, ultimately gave the final protein which did some effect, and then it cascade keeps going on. So whenever we read immunology, there are so many pathways and we get screwed up. कि यार इतना नहीं पढ़ना है. I can't remember this. And this is even statement today. But it is little different. इतना पढ़ना है, याद नहीं रखना है. Okay. So then immunology becomes quite easy. If you are reading, not trying to remember too much, you will actually understand. And then things will be, we can understand. Now see, this looks so complex, right? But what I'm trying to tell here is there are so many cytokine which is working through JAK-STAT pathway. Okay. So if we have JAK inhibitor, do we have JAK inhibitor in market? Yes or no? If yes, please name few JAK inhibitor. Anything which can come on the message, I'll be happy. Okay. So those JAK inhibitor, when they're given, it can actually do the function. The problem at this point, okay, we got tofacitinib, baricitinib, roxolitinib, right? So this all are things which are JAK inhibitor, which if affects this can lead to decreased signaling from this pathway. So that disease is taken care. But when we are actually reducing signals from multiple pathway, there is very high chance of developing infection. So whenever we prescribe JAK inhibitor, your disease gets controlled very well because you are targeting multiple targets through that. But your chance of infection susceptibility goes high for sure. And in that, varicella zoster virus or the shingles are quite active, which becomes so zoster activation is quite common after this use of this. Do you know any other drug which can work on multiple pathways like this? Any other drug which can see JAK inhibitor has this beauty. It can target multiple cytokine together. IL-6 is also targeted. Interferon is also targeted. IL-4 is targeted. 9 is targeted. TNF is targeted. Everything is targeted together. Do you know any other drug which can actually do this massive inhibition like this? We all have used that drug. Any guess? Steroids. Okay, steroid does that. No, HCQ is quite innocent compared to this. Okay, so when we give HCQ, we are not much bothered, right? Ki patient will develop too much infection because immunity is affected. No, but when we use JAK inhibitor, when we use steroids, we are always concerned about infection because they are non-specific inhibitor of multiple pathway. So for both the drugs, the response is amazing in autoimmune or inflammatory disease, but it comes at the risk. Okay, that's what we have to remember, that it comes at the risk. So we have to be little more careful, though they are very common in use, both. Okay, steroid also, JAK inhibitors also. So now this is what the end comes. The Harrison has given table where they have named the cytokine, given the receptor, given source of that, given target of that, and given function of that. Why I was repeating this multiple times, do things at your own pace. Life is not a race, because I actually wanted to show you the tables of cytokines which are given in our Harrison. Okay. This is to be read once. It is there in your Harrison. I'm not going to discuss, otherwise class will never going to get over, but you know or you understand the principles. After that, just try to get overview. Do not go in too much detail. Sometime I will actually sacrifice on one MCQ rather than screwing my gray matter so much. Okay. And then there are innate cells. I will discuss this part in the next few classes probably. Okay. So that will come to this, and that comes to the end of innate immunity. My next class, what I will plan will be more on adaptive immunity, lymphoid organ, and then the hypersensitivity reaction type, completing the immunology series. I hope you are at least understanding what we are discussing. Sometime I don't believe in much levels of discussion, but I wish that whatever we are discussing makes sense and it is worth discussing point, right? So anything worth having will take time, and it will take its sweet time. I think 4 or 3 more lectures, I'll be done with the immunology series. Thank you for your time, and we'll end our class here. Any input you can tell us. More than happy to receive.